I want you to understand all of the factors that contribute to the effectiveness of a method of control. It's not just the type, but the number one reason is the type of microbe. And the scale from easiest to control to most resistant to means of control, as far as what types of microorganisms are the most challenging to control. And we should already have an idea of that from the results of our disinfectant experiment. We'll see an idea of that today or this week when you look at the results of your effectiveness of disinfectants. But we know something basic, one basic difference between the two bacteria that we tested in that experiment is what? As far as their features or the capabilities. So we tested two bacteria, E. coli and Bacillus subtilis. And Bacillus subtilis has an additional capability that E. coli doesn't have. What is that? Endospore formation. Okay, so it's much more difficult to control endospores than it is to control bacteria that don't form endospores. Also, in general, that extra lipid bilayer that gram-negatives have make it a little more resistant to most chemical disinfectants versus gram-positives. Okay, so, and there are also variables depending on the effectiveness of the means of control that you're selecting, how long it takes to have a microbicidal effect, or how toxic it is to the environment or to the surface. So we're restricted, for example, when we want to clean a tissue like a surface of our skin before we insert a needle. We're restricted by certain properties of the chemical we're using in disinfection, obviously, because we don't want to damage the skin, the live tissue. Also, some disinfectants are sensitive to the presence of organic materials, things like bodily fluids, blood, or they don't penetrate very far. So things like non-ionizing radiation is used, but it doesn't penetrate very far beyond the surface. So this is things like UV light, like sunlight. It won't penetrate far into a substance, so it only really helps in removing the microbes on the surface of materials. Also, the material of the item that you're trying to sterilize or to reduce the amount of microbes on. So a plastic would be sensitive to sterilization by heat, obviously. So other methods must be considered. So there are several variables that you need to think about when selecting a procedure, whether it's physical or chemical. You should understand the microbial death curve and the D value, the time that it takes to reduce the population by 90%. And there are varying degrees of control. So as I have been talking about, sterilization is one of the most complete means of control, in that it removes 100% of the microorganisms present on that item or on that surface. But there's a range of controls. We don't always want to sterilize everything. We don't have to sterilize our food. We just have to reduce the number of microorganisms so that it's safe for us to consume. And so there are different degrees of control, all the way from not any specific means of control, just cleaning that area, but no measurable means of control as far as sanitizing versus 100% of the microbes removed in the case of sterilizing. There are four major areas of physical means of control: heat, radiation, filtration. The final one being mechanical removal, so that's like washing our hands. And mechanical removal can be aided by chemicals, so we use soaps. These are generally chemicals that are not affecting the growth or the viability of the bacteria. They simply aid in the mechanical removal. So soaps are not killing the bacteria unless they have some added chemical to them, such as triclosan or something that will be antimicrobial in its activity. But just your over-the-counter soap helps to break up the oils on the surface of that build up on the surface of your skin. They help to remove those kind of dead layers of skin that are that will slough off eventually if you just use water. So some of you found differences between using soap in addition to water, right? An improved removal of the bacteria on your fingers, whereas others of you found that there really wasn't a big difference between water washing versus washing with soap. Okay? And that's really because the mechanical rubbing and scrubbing has a lot more to do with the effectiveness of hand washing than actually just putting on a little bit of soap and rinsing off your hands. Okay, the agent used. The soap will aid in that because it's able to break apart the oils and better remove things. But it's not killing any microorganisms or destroying any viruses on the surface of your hands. It's simply aiding in the removal of them. So chemical methods include metals and disinfectants and surface acting substances like soaps or quaternary compounds. And the chemical, just like the physical methods, depends on the situation that you're faced with, that you want to control, in which you want to control the microbes, and the degree of control that you want. And there are slightly different terms used for disinfectants. But in general, whether it's a chemical means or a physical means, to kill a microorganism, or in the case of viruses, to destroy or inactivate the virus, the suffix "cidal" will be used. So virucidal will be something that refers to an agent or a method that inactivates or destroys viruses so they can no longer infect a host cell and replicate. Now there's no term virustatic because viruses are what? Non-living. But in the case of other members of the microbial world, like fungi, there's fungicidal agents or methods and fungistatic methods. So once the fungistatic agent is removed from the fungi, it will start to grow once again. So this is a temporary control that reagents that are static in limiting the growth of the microorganisms. So what would the terms be for bacteria in the case of limiting the growth of bacteria? Bactericidal would actually kill the, you're right, bacteriostatic, yes. And there are different degrees of killing that you can achieve. You can with the method that would achieve sterilization, that's the removal or the killing of 100% of microorganisms. Filtration, heat, chemicals, and irradiation can be successful at sterilizing either liquids or instruments or solid material like glassware used in our laboratory, for example. Disinfection gives a measurable amount of reduction. 90% of the pathogens or the microbes are removed, but it doesn't give a complete elimination of the microorganisms. And in the case of chemical disinfection, disinfectants can give this level of control, as well as some antiseptics. But usually antiseptics, it's not a measurable degree of control. Now, one of the ways that we can control microorganisms through the use of heat is pasteurization. So this is very high heat for a short period of time. It doesn't give any measurable reduction in the or any specific reduction in the amount of microorganisms, but it reduces them to a safe level. So while we don't know whether it's 50% reduction or 60% reduction, we know that it's reducing it to a safe level that slows the process of food spoilage. Eventually, the milk or the juice or the cheese or whatever's being pasteurized will spoil. And there are other means that we use, like reduction in temperature, to extend that shelf life and reduce or slow the spoilage. But it's not a measurable amount of reduction, a set amount. The same thing with these other means of control. They're not a reproducible reduction in pathogens, but they reduce the pathogens to the level that's considered safe. So decontamination is reducing pathogenic microorganisms. So it would affect all microorganisms, but our concern more here is the level of pathogens among all microorganisms to a safe level. Degerming just refers to reducing the number of microbes on tissue, like skin. Sanitizing is reducing the population of microorganisms, but it may not necessarily be considered a safe level, as far as it still may be, the pathogens still may be present. And preservation, again, to delay the spoilage. So there's still those microorganisms that contribute to the spoilage of food present, but they're just at a lower level, or their growth is inhibited such that the spoilage will be delayed. So things like desiccating, drying out food, freezing your food, adding salt, salting meats or smoking meats, adding chemical preservatives, BHT, and things that you see in the ingredients of your foods in the supermarket. Now, the situation, so the level of control is one thing to consider when you're choosing a method of control, but also the situation is important. You may not be so concerned about reducing the level of microbes in your daily life, except for maybe in the kitchen. But when it comes to hospital disinfection and means of control of microorganisms in the hospital, there are entire panels and groups dedicated to the constant monitoring of pathogenic bacteria in hospitals and promoting means of control of these pathogens. And okay, and this is just a list. I'm going to go, I have it already in the following slides. So microbiology laboratories, we've already used the aseptic technique. That's one example of how we want to control the environment so that we're just studying that pure culture of bacteria that we're interested in and not any other bacteria in the environment. Food and food production, obviously, you have to understand these death curves in order to design a proper procedure for canning foods, for example, the proper temperature and time that the food has to be exposed to that high temperature in order to successfully remove the microorganisms. And even water treatment. So clean water is one of the most effective ways at reducing the spread of disease. And before we had water treatment systems and a system of delivering water to the community, diseases like polio and outbreaks like of bacterial infections like cholera were not uncommon here. But because we have a very tightly regulated water supply and an effective means of treating water, that helps reduce the degree of infectious diseases and the spread of infectious diseases within our communities. Okay, so as I mentioned, when selecting the procedure, the most important factor is the type of microbe that you're targeting. So, for example, if you simply want to reduce the number of bacteria, so washing our dishes, washing our hands, washing our food is a very effective means of reducing bacteria on our food. But if you're more concerned about things like endospores or prions, so a surgical instrument, for example, is going to go beyond these barriers that we have of our skin and mucous membranes. It's going directly into areas of our body where there are no microorganisms present, but there are plenty of nutrients present to support this growth. So in the case of a surgical instrument, you want to, or controlling endospores and prions on a surgical instrument, you need much more rigorous means of control and a different procedure. Obviously, the amount of the microorganism there is going to influence how you design the procedure. Okay, so the more bacteria you start out with, the longer it is for the chemical to control it. Often, simply washing the item before you use your means of control is a very effective way at reducing the extent of contamination so that the treatment procedure, for example, in chemical disinfection, doesn't need to be as long. Or even in the case of heating items like autoclaving, heating with moist heat, a steam at high pressures. If you wash the item beforehand, you're reducing that level of contamination so that a much shorter cycle is necessary to achieve sterilization. The environment and the composition of the infected item kind of go hand in hand. The materials that the item is made of, is it liquid? Is it an electrical instrument that would be sensitive to liquids or hazardous if you used a liquid means of disinfection or sterilization? Are they plastics? Are they food items that would be you want to maintain as fresh? So obviously you can cook the food item, but there are means of controlling microbes on things like meat that doesn't involve heat but will reduce the amount of microbes on the food. Environment also, you want to be sure that the means of control is going to be safe for the environment, that you're not going to produce any byproducts or leave any residues that may be harmful to the environment. And then the risk of infection. So in your kitchen, you're not really worried too much about the risk of infection because we're assuming that you're a healthy individual, you're not in the hospital, you're not immunocompromised in any way. But when you're considering a site like the hospital, almost all of the patients in the hospital are immunocompromised. So having that weaker immune system, it's much more difficult for them to fight off infections, so the means of control has to be much more rigorous. Also, close quarters, crowded conditions like a dormitory would necessitate different procedures maybe than uncrowded conditions. Okay, so why is the type of microorganism so important? Well, this is because of features we already talked about, at least with bacteria, and we'll soon be talking about with other members of the microbial world. But remember, endospores are these very dormant, very resistant forms of stages for bacteria. And they have many layers that protect them from UV irradiation, from chemicals entering. So a lot of the chemical disinfectants cannot penetrate these many layers. Also, they're resistant to drying out and resistant to high heat. So even boiling water for hours may not remove the endospores in that solution. So in the case of more resistant microbes like endospores or prions, which have no susceptibility to, so prions, remember, are these infectious proteins. Usually proteins are broken down by enzymes called proteases, but prions are resistant to protease action. So these are some very resistant microbes, very hard to control. And I'll go over how autoclaving is very effective at controlling them. So moist heat, steam, with the addition of pressure. Also, we talked about the difference between Mycobacterium having this extra layer of mycolic acid in their lipid bilayer beyond their peptidoglycan. That makes it more resistant to the entry of substances, including disinfectants. And then these more dormant forms of life, just like bacterial endospores, there are spores of fungi, and protozoa have a spore-like stage of their life cycle called cysts or oocysts that are more resistant to the penetration of chemicals or more resistant to high heat. In general, gram-negatives are more resistant than gram-positives. And any actively growing microorganism, whether it's fungi, algae, protozoa, bacteria, if they're actively growing, they're more susceptible than those that are not. And then as far as viruses go, they tend to be less resistant, but they're more susceptible if they have an additional layer around just the nucleic acid content and that protein coat that makes up all viruses. So naked viruses means just they have this core structure of nucleic acid in a coat of protein. But some viruses, especially human viruses, have an additional layer, a lipid bilayer. And those are called enveloped viruses. That lipid bilayer makes them more susceptible to control. Bio what? Biofilms, right, yeah, communities of bacteria, yes. Yes, I'm just talking about individual types as well, but yes, any time that you have a microorganism that's within organic matter, like in a cut, the most important thing before you try to disinfect is to clean the area. Okay, so dirt, for example, can inhibit the effectiveness of a disinfectant like hydrogen peroxide. Okay, so a lot of these organic materials will inhibit the function of chemicals used in disinfection. Also, things like quaternary compounds that are they're only effective on surfaces. Yes, Letitia. Right, yeah. Heat and a combination of heat and chemicals is what's used to destroy prions. And simply other means like oxidation. Okay, so in the lab when we flame our loop, we're essentially oxidizing everything there to complete end. So we're just incinerating everything that's there. Yes.
Actively growing, do you mean like in the log phase of growing, or is that?
Yeah, at least log phase or stationary phase where the majority of the organisms are growing, yes. So chemical disinfection is not as effective against endospores. They're usually controlled by this process of autoclaving, which is steam. So moist heat is much more effective than dry heat because it penetrates the item that you're trying to disinfect and heats it up much faster, as well as pressure. So autoclaving is a combination of steam and pressure, so moist heat and pressure. Prions, incineration is the most effective way to deal with prions. So a lot of disposable items are used when coming in contact with tissues. Okay, so these would be neurological tissues, brain and spinal cord tissue, because of the site of the infection of prions. So whenever possible, the items are just disposed and incinerated. But in the case of those items that need to be reused, autoclave for several hours at a higher temperature than a standard run. So standard run for autoclaving is 121 degrees for about 20 minutes. I think it's 15 psi of pressure for about 20 minutes. But as you can see here, 4.5 hour run, that's a very, very long run for the typical autoclave. You can autoclave liquids or solids. So glassware, or like the broth that we use in laboratory is autoclaved. But in the case of prions, materials with prions, it requires this higher temperature for a much longer time. Also, if you have liquid waste containing prions, you need to use the combination of chemicals like very concentrated sodium hydroxide, then followed by autoclaving. Some other chemicals are also used, phenols and hydrochloride, guanidine hydrochloride or isocyanate. But most commonly, the concentrated sodium hydroxide is the most effective. And it requires a very long time for these chemicals to be effective in destroying the prion. So here's the next variable, the extent of contamination, how much of the microorganism is present, is where you need to understand the nature of the death curve, how bacteria, for example, will die, at what rate. And this curve can change depending on the organism, depending on the temperature or the pH that you're treating it at with that chemical, the concentration of the chemical. But this is just to show you that the time that it would take to kill For example, I think this is E. coli being treated with bleach, a chlorine-containing, a halogen-containing compound. So this time that it takes in order for the microorganisms to die is measured on a log scale, and here's the time, and it's called the D value. And so any time that you go down one log order, so here they're measuring 1,000 reduced to 100. The difference between that one log value, one D value, is about 20 minutes or so, or yeah, about 20 minutes in this case. Okay, so that's going to be different for each, the slope of each organism that's measured. So for example, here, if I have, and here, because it's a log scale, you're reducing the population by 90%. You still have 100 organisms left, but from each log is a measure of 90% of the population. So if you reduce the population by 90%, but you're starting out with 1 million bacteria here at 10 to the 6th, you're still going to have a lot of bacteria left after just one D value reduction. So in the case of a population where you have 10 million, for example, and you want to reduce it to just almost nothing, like 10 organisms, 10 to the 1st power, you're going to calculate it by taking the exponent, okay, and you have 10 million, you want to go down to 10. So here, 7 minus 1, okay, so you need to go 6 D values, or 6 90% reduction of the population to get down to this point of having 10 organisms left, right? And then the D value, if the D value equals 10 minutes, how much time would that require for this specific chemical, pH, and temperature? So it would take 1 hour, or 60 minutes. Okay, so that's how you calculate the time that it would require to reduce the bacteria to a certain point, a certain number. Each D value is only a 90% reduction. So the more you start with, the longer time will be required to get down to a safe level. And that's called, the D value is called the decimal reduction time. It's an actual measure of how much time it will take at certain, so this doesn't tell you, but it would be different depending on the temperature where you do disinfection. So the chemicals are a little bit more effective at higher temperatures, many chemicals, but they tend to be most effective at neutral pH or near neutral. It's not true across the board, but in general. So at a fixed temperature and a fixed pH, and you're talking about a single type of bacteria, so this would be different if I was talking about Bacillus subtilis, for example. The killing time would shift. Okay, so the line would shift into, so here's E. coli, and for Bacillus it would shift up. So it would take a longer time to reduce the same amount of bacteria because it has this feature of endospore formation. Same thing with gram-positive versus gram-negatives, because gram-negatives have this additional lipid bilayer around their peptidoglycan. Okay, so environmental conditions I already touched upon. The temperature is going to influence the effectiveness of a procedure, or especially in the case of chemical disinfection, pH influences the presence of organic materials. Things like quaternary compounds may not be as effective when they're used in a bandage, for example, that's saturated by that cellulose-containing material, decreases the effectiveness of the chemical. In general, chemicals do better at higher pHs and neutral or lower than neutral, higher temperatures and neutral or lower than neutral pH. And when I say organic materials, I'm talking about anything like dirt, grease, oils, bodily fluids like blood. All of those things can have an influence on some chemicals. Other chemicals work effectively in the presence of these organic molecules, but some are affected, like hydrogen peroxide would be one of those affected by the presence of organic material. And as I mentioned, the risk of infection would also determine what procedure is chosen. So in the case of the medical setting, there are certain items that are critical. You know they're going to go in contact with bodily tissues so surgical instruments, for example, that will penetrate our own natural barriers to infection. Semi-critical instruments would not go past the skin or mucous membranes, but they might be introduced into the body. So, for example, during a colonoscopy or a catheter, for example. And non-critical instruments, things like a stethoscope, would be a non-critical instrument. So little risk of transmission. So clearly, the critical items have to be sterilized. 100% of those microbes have to be removed in order for it to be safely used. The composition of the item, as I mentioned, yes? It comes in contact with a mucous membranes. So, for example, a urinary catheter would not be breaking through the mucous membranes, but it would be inserted into the body, into a body cavity.
So a critical item would be like a pacemaker or something, right? What would be?
Yeah, any kind of artificial or mechanical instrument that would be implanted into your body would be a critical item. Yeah, it had to be free of microbes. Does that make sense? Anything coming past those barriers. So sometimes in certain procedures there are tubes inserted down your throat, so that would be a semi-critical item because it's not actually penetrating into the tissue beyond those mucous membranes. And then the critical items are a break in the skin or body tissues. Something for a pap smear, it would be a semi-critical, but usually sterilized instruments are used, yeah. I mean, sterilized swabs and things like that because you need to be just measuring what you're sampling and not some other microorganism that may have sat down on the instrument before you used it. So that's a little bit different reasons. So the risk of infection is semi-critical, but to successfully administer the test, then you would need to have it be sterile. Okay, so I already mentioned a little bit about the composition of the item. Obviously, if something is corrosive, you're not going to use it on a metal. If it's high heat, it's not going to work on plastics. Toxicity is a concern when you have the skin as a surface that you're trying to disinfect or degerm. And then waters or liquids may be heat sensitive, have heat sensitive components, and may need some alternate means of control, like a filter. So, as I mentioned, the physical approaches to control include this heat. Autoclave and pasteurization are the ones that I want you to understand for heat. Irradiation, there are two types. There's ionizing and non-ionizing. So things like X-rays are ionizing. Sunlight is non-ionizing. The filtration of, sorry? Oh yes, for irradiation there are two types. There are X-rays and gamma rays, which are ionizing irradiation. And there's UV light, like from the sunlight, which is non-ionizing. Filtration is the physical separation. So there's tiny little holes in these filters that can, based on the size of the microbe, reduce or eliminate any bacteria, for example, from that solution. It's usually used for liquids. There are filters that are small enough to reduce or eliminate any viruses in the solution. Also, filters are used for air in the case of critical situations where you want to remove microbes from the air. In the case of high efficiency particulate air filters, these HEPA filters. And then the mechanical removal I already talked about. So the key difference when we're talking about heat is that there's moist heat or dry heat. Dry heat isn't as effective as moist heat, like steam. So think about when you put your hand in the oven, it feels hot, but you don't get burned. But when you put your hand above that boiling water, ouch, you're out of there right away. That is because it's the heat, it penetrates your tissue much faster. And heat in general, whether it's dry or not, it destroys the organisms by destroying the proteins. So denaturing or coagulating the proteins so the enzymes can't work anymore. So there's boiling, which is very effective at destroying most microorganisms and viruses. If you boil long enough, you can eliminate endospores. But it is not always effective against all endospore forming microorganisms. So in that case, you need to use the moist heat in addition to the pressure of the autoclave. Pasteurization does not sterilize. It doesn't even reduce the organisms by a fixed amount, but it does increase. It reduces the organisms enough so that it increases the shelf life. And there are two types of pasteurization. There's high temperature short term, HTST. So a 15-second treatment at 72 degrees, or there's ultra high temperature, UHT, where it's way up around 150 degrees, but just for several seconds. So these are very short-term treatments, but very effective at reducing the microbes that would spoil the food and that would make us sick. Pressurized steam is the autoclave. This is the machine that's used to prepare all of our laboratory supplies. It can sterilize, very effective means of sterilizing. Sterilizing metal instruments, glassware, liquids. And the standard run is 121 degrees, 15 psi. I think your book says 15 minutes, but 15 to 20 minutes is usually sufficient to sterilize anything on a piece of glassware, and metal instrument, or a liquid. And it's effective against these short standard runs are effective against endospores. But remember, they're not effective against prions. Prions require much longer autoclaving and higher temperature, same pressure.
So PSI is pressure.
Yeah, PSI is the pressure, yeah. Pressurized steam, yeah. Other physical methods, so I mentioned liquid filtration based on these pores, but there's also air filtration. This is important when you want to control the microbes going into, for example, from this pump, there might be a HEPA filter that prevents the introduction of any microbes into the solution after it's been filtered. Or in the case of critically ill patients that are susceptible to respiratory illnesses, there may be HEPA filters controlling the room. Also, things like negative pressure would be used to prevent the air from the outside of the room coming in. And the air systems in hospitals are highly monitored and controlled, and filters are an important source of protection. Even the filters that you use in your AC or your furnace, if you have hot air, heat in your home, are an important way of preventing infection. Legionnaire's disease is an example of one disease that was spread. It's actually named after the initial time when an outbreak occurred at a Legionnaire's convention because the AC filter was not clean. And this bacterial respiratory infection affected almost everybody who attended that meeting. So it's really critical to have these filters removing the microorganisms in the environment. I already went over ionizing radiation, an example, X-rays. These radiation is not affecting the proteins, it's affecting the DNA. So think about the risk of going out in the sun in the summer. You might get sunburn. The changes that occur in your skin over time from repeated exposure to this sunlight, which is the ultraviolet radiation, changes your DNA such that it can cause cancer. So cancer is initiated by changes in your DNA. And so radiation, whether it's ultraviolet radiation or ionizing radiation, it affects the DNA of the microorganisms. Without functional DNA, they can't replicate.